Hi everyone, thanks for tuning in to this advanced hoof care seminar, uh, The Path to Obesity-Related Laminitis. I'm Bruce Nock. It's Thursday, August 8th, 2024, and it's a beautiful day here in Missouri. So today I'm going to tell you how eating too much puts a horse at risk of becoming laminitic, an extremely painful condition that results from the inflammation and weakening of the sensitive tissues outlined in red that attach the coffin bone to the hoof wall. So a horse overeats, becomes obese. That leads to inflammation, insulin resistance, and too often laminitis. Specifically, I'm going to tell you about how and why these pathologies occur. This is a dynamic topic. Things change as we learn more about how these pathologies develop and progress toward laminitis. And the really complicated processes that involve the interaction of literally hundreds of signaling molecules, enzymes, immune cells, receptors, etc. Obviously, I can't cover all the details, and frankly, you wouldn't want me to. But I'll try to give you the gist of it in understandable terms as we know it today. That should give you a foundation for talking with veterinarians and nutritionists about these pathologies and for making decisions about how to treat them, and more importantly, prevent them from developing in the first place. So let's get started. Inflammation is where things start to go wrong. Inflammation is part of the innate immune response. It's one of the first actions the body takes toward neutralizing potentially harmful material resulting from damaged or dysfunctional cells, pathogens, irritants, cuts, what have you. Wounds and infections don't heal without inflammation, but too much of a good thing can be harmful. Inflammation that goes on for weeks, months, or indefinitely is a pathological condition that slowly destroys the tissue it was intended to protect. Even worse, it's now known to be the cause of a number of major degenerative conditions, including insulin resistance, which I'll talk about shortly because it's the important next step in the progression toward obesity-related laminitis. So where does obesity-related inflammation come from? Obesity isn't a wound or a virus or a bacteria or any other thing that historically are known to trigger an inflammatory reaction. Well, not so long ago, scientists unexpectedly found that food induces a very mild inflammatory reaction, or more precisely, the metabolic processes that eating trigger induce a very mild inflammatory reaction. Don't ask me why food does that because I haven't heard a satisfactory explanation yet, but it's likely to involve the activation of inflammasomes, an intracellular protein complex that regulates immune and inflammatory responses. Now, in lean animals, the food-induced inflammation dissipates during periods when they aren't actively eating. In other words, the inflammation follows the eating cycle, as shown by the blue line, with peaks associated with eating and valleys associated with fasting. And over time, the food-induced inflammation stays at a low baseline level. But when an animal overeats, the inflammation doesn't go away completely during periods of fasting. So the inflammation keeps building on itself, getting worse, as shown by the red line. So that's how overeating leads to inflammation. Now, Acute inflammation is a good thing. Wounds and infections, etc., don't heal without it. It's the body's natural remedy for such things. The problem is obesity leads to chronic inflammation. Obesity-related inflammation essentially goes on forever. Why? Why is obesity-related inflammation so persistent? Well, I already mentioned one factor. When a horse overeats, the inflammation doesn't go away completely during periods of fasting, and so it keeps getting worse, building on itself each time the horse eats. But there's another important reason why obesity-related inflammation doesn't resolve, and it explains why, in addition to increasing the risk of laminitis, it makes all sorts of other inflammation-related conditions worse, like arthritis, autoimmune diseases, etc. Obesity compromises the ability to turn the inflammatory reaction off. You see, until recently, it was thought that triggers for the inflammatory reaction, like bacteria, viruses, etc., would in time be removed as a consequence of the inflammation itself. And so it would just stop. There'd be nothing to keep it going. Stop putting wood on a fire and it goes out. That's how inflammation was envisioned. That turns out not to be the case. It's much more complicated. Inflammation doesn't just stop. Resolution is orchestrated by an army of signaling molecules. It's sort of like driving a car. To stop it, first you take your foot off the gas pedal, or in this case, the case of inflammation, remove the trigger. Get rid of whatever caused the inflammation in the first place. Then you apply the brakes. Applying the brakes to inflammation is the job of specialized pro-resolving mediators, SPMs. They put the brakes on inflammation. They don't suppress or block it. They signal for it to stop. The problem is, it now looks like brake failure is an important contributing factor to inflammation-related disorders in humans, like cardiovascular disease, osteoporosis, arthritis, depression, Parkinson's disease, COVID-19, and insulin resistance. It's very likely to be the same for horses. You see, obesity actually drives an SPM deficiency. It compromises the braking system. It's like trying to stop a car with worn out brakes. Bad news. The car just keeps going toward that inevitable collision, and so does inflammation in obese individuals. But in this case, the collision is with some sort of health problem. Obesity makes health problems worse because it undermines the recovery from the inflammatory reaction itself, and the damage caused by its sustained activation deepens. It can damage cells, tissues, and organs. It can cause internal scarring, tissue death. It can even damage DNA. Ultimately, it can lead to the development of disabling or life-threatening disorders, like insulin resistance. So now you have an idea about how obesity-related inflammation develops and becomes chronic. The next question is, how does inflammation lead to insulin resistance? Let's start here. Horse feed, grain, and what have you, is composed of these 7 basic components. I'm going to focus on carbohydrates. I'm not a nutritionist, so I'm not going to tell you how much a horse should eat, the difference between structural and non-structural carbohydrates, and that sort of thing. There are people who are much more qualified than I am to talk about such things, and there's plenty of information on the internet. I'm more interested in what happens to carbohydrates after they're digested and how it leads to insulin resistance. Let's start at the beginning. Digestion degrades carbohydrates to glucose, the lead character in this segment about insulin resistance. Glucose is a vital energy source for all mammals. It's the primary metabolic fuel for horses, other equines, and ungulates in general. Once inside the body, glucose travels through the bloodstream to tissues that need energy. When it gets inside the cells of those tissues, it's converted to adenosine triphosphate, ATP. ATP provides the energy to drive and support many processes in living cells. It's the gas that drives the cellular engine. Here's the thing. Glucose needs help to get into certain cells. Insulin provides that help. Here's how it happens. Carbohydrates are consumed and broken down to glucose. The glucose is then absorbed into the bloodstream from the digestive tract, like every other nutrient. As a result, the amount of glucose in blood increases. That triggers the release of insulin from the pancreas. Insulin then travels through the bloodstream and tells fat, muscle, and liver cells to make it easier for glucose to enter them. It delivers that message by attaching to specialized molecules called insulin receptors on the surface of those cells. Here's a visualization of the process. This little squiggly looking fellow is an insulin molecule traveling through the bloodstream. The cell membrane is that yellowish brownish structure running across the slide. And the two purplish things is an insulin receptor which is embedded in the cell membrane. The receptor is dormant, not doing anything until insulin activates it. That sets off a series of chemical reactions inside the cell. It's called the signal transduction cascade. As you will see as we go along, it's the key player in the development of insulin resistance. It does two things. It amplifies the insulin signal, so the signal from an insulin molecule doubles and then quadruples and so on. The signal transduction cascade also inserts molecules called glucose transporters into the cell membrane. The glucose transporter provides channels for glucose to pass through. So let's review. Carbohydrates are degraded to glucose. The glucose is then absorbed into the bloodstream. As a result, the amount of glucose in the bloodstream increases. That triggers the release of insulin from the pancreas. The insulin activates insulin receptors on the surface of fat, liver, and muscle cells. Not all cells, just fat, liver, and muscle cells. The activation of those receptors sets off a cascade of chemical reactions inside the cell that amplifies the insulin signal and promotes the proliferation of glucose transmitters on the cell surface, which provide channels for glucose to pass through and into the cell. Then the intracellular glucose is converted to ATP, which provides energy for the cell. That's how it's supposed to work, and how it works in a horse that is in insulin resistance, has a normal sensitivity to insulin. So what goes wrong when a horse gets obese? Well, this is where our story about inflammation and insulin resistance comes together. Remember this slide? The red line shows the inflammation keeps building on itself when a horse eats too much. As the obesity-related inflammation progresses, immune cells jump into the action. Immune cells are a normal component of the inflammatory reaction. It doesn't matter whether the inflammation is sparked by a wound or, in this case, overeating. It follows the same course of action either way, and neutrophils are typically the first immune cells to react. Neutrophils are a type of white blood cell. They're formed in bone marrow and travel to where they're needed through the bloodstream, tissues, and lymph nodes. Neutrophils do two things of importance to our story. First, they secrete an enzyme called neutrophil elastase, which impairs insulin signaling and decreases insulin sensitivity to some extent. Second, they summon another type of immune cell called macrophages to the site of inflammation. As macrophages proliferate in fat tissue, they release a number of protein messengers called cytokines, some of which, like tumor necrosis factor alpha, make cells insulin resistant. The body fails to properly regulate, use, and store glucose. And that is how chronic inflammation causes insulin resistance. So here are the major steps I've covered to this point. Take in more calories than you burn, and you gain weight and eventually become obese, which leads to a persistent subclinical inflammation. The inflammation activates neutrophils, which release neutrophil elastase and induce a degree of insulin resistance. The neutrophils also recruit macrophages to the site of inflammation. The macrophages released cytokines, which cause cells to be insulin resistant. And here are the details of what happens between the release of the cytokines and insulin resistance. The cytokines activate intracellular kinases that phosphorylate serine residues in the insulin receptor substrate 1. That impairs the insulin transduction cascade, and that is the key event that causes insulin resistance. And that's basically how eating too much leads to inflammation and subsequently insulin resistance. Again, a condition in which the body fails to properly regulate, use, and store glucose. So why can't the body process glucose in the normal way? Because it can't move it out of the bloodstream and into fat, muscle, and liver cells as it normally does. So the remaining question is, how does this chronic obesity-related inflammation that causes insulin resistance increase the risk of laminitis? Well, despite what you might have heard, the short answer is no one knows at this point. But there are some things we know that might provide clues. One is that insulin resistance causes high blood levels of insulin. And at this point, I think most people believe that's what causes laminitis, the high levels of insulin. In fact, it seems like a lot of people, including veterinarians, have written it in stone. For example, here's what Dr. McFarland has to say about it.
We have a few people asking about the connection between insulin resistance and laminitis. Can you expand a little bit on that and how they can go hand in hand?
So there's some very good evidence that we've learned probably in the last 15 years that insulin itself directly causes laminitis. So you can take a perfectly normal animal, and if it gets delivered high concentrations of insulin, within 12 hours you can start to see changes in the feet. And by 3 days, they're actually sore-footed. And so we know that insulin directly causes the change of laminitis.
Okay, so most veterinarians would agree with Dr. McFarland, and I agree with her to a point. A sustained infusion of a high concentration of insulin does cause laminitis. But it's a big leap from there to saying that we know laminitis is directly caused by insulin. That conclusion would never be allowed in peer-reviewed journals, at least not the journals I've dealt with. There are just too many gaps in our current knowledge. A major problem is we don't know how infused insulin causes laminitis. Probably the most prominent and widely accepted theory came from Dr. Pollitt's research group in Australia. This is how they conceive of it happening. The high levels of insulin that normally go along with insulin resistance activate IGF-1 receptors, insulin-like growth factor receptors, on the Miller cells in the foot. That in turn causes the proliferation of laminar epithelial cells which stretch and weaken the lamina. Then the weight of the horse breaks the bond between the external hoof wall and the internal structures, and voila, you have laminitis. Maybe so, it's possible, but I'm not totally convinced yet. And here's why. All of the evidence and conjecture is based on findings from the infusion of a high level of exogenous insulin, administered insulin, not endogenous insulin, insulin that's actually made by the body. This is really important. There are lots of examples showing that administered hormones and hormones produced by the body itself have different effects. For example, here's a study that reported different effects of endogenous and administered insulin, and I'll let you read. So in simple terms, this study found that administering insulin makes people hungry, but the insulin made by the body does the opposite. It decreases hunger, makes people feel full. So showing that the infusion of insulin causes laminitis is a valid first step, but it doesn't establish that the insulin produced by the body itself does so. This is a big deal, a huge gap in the available evidence, not just some scientific detail. So I'm not dismissing the possibility that the high levels of insulin might cause laminitis. It's possible, but for me, the jury's still out. I'll wait for more data before writing it in stone. Here's another problem, and again, I'll let you read. So for me, even if endogenous insulin does cause laminitis, the question of how it does so remains to be established. Apparently, according to the findings of this study, it doesn't do it as Dr. Pollitt's group suggested. Okay, moving on. So the thing that puzzles me most about the cause of obesity-related laminitis is the tunnel vision on insulin while glucose has been almost totally ignored. This is really weird to me because the damage caused by type 2 diabetes, severe insulin resistance in humans, is caused by glucose, not insulin. Glucose gets into cells in two ways. One is insulin-mediated glucose uptake, which is what I've talked about so far and that gets compromised in insulin resistance. It occurs in insulin-sensitive tissues, specifically liver, muscle, and fat tissue. The second type of glucose uptake is non-insulin mediated glucose uptake. Insulin isn't required. Whether insulin levels are high or low makes no difference. It's the amount of glucose in the bloodstream that's important. In this case, the glucose gets into cells by what's called facilitated diffusion. It occurs in both insulin-sensitive and non-insulin sensitive tissues. During fasting, approximately 80% of glucose uptake into cells actually occurs by this insulin-independent mechanism. Now, when an animal becomes insulin resistant, a number of significant things happen. One, the amount of insulin secreted increases to compensate for the decrease in sensitivity to it. Two, glucose levels in blood increase because its entry into insulin-sensitive cells is decreased due to the drop in insulin sensitivity. That means more glucose stays in the bloodstream because it can't get into liver, fat, and muscle cells. 3. Glucose levels in non-insulin sensitive tissues, tissues that don't require insulin to take up glucose, increase because there's more glucose available in the bloodstream for absorption. And that's why non-insulin sensitive tissues are at risk for glucose damage when an animal or human is insulin resistant. And as insulin resistance gets worse, more glucose gets into those cells. Now let me back up for a minute and put things in perspective. The focus on insulin rather than glucose emerged from an early study that found that, in contrast to insulin, infusion of glucose for a few days doesn't cause laminitis. The thing is, short-term elevations of glucose aren't harmful, but excess glucose hanging around in the bloodstream or flooding tissues that don't depend on insulin for glucose uptake is poison. So it isn't surprising that in humans, the tissues and organs that aren't dependent on insulin for absorption of glucose, like the kidneys, blood vessels, peripheral nerves, and lenses of the eye, are primary targets for damage due to insulin resistance. It's not damage caused by insulin. It's damage caused by glucose. And guess what? The hooves of horses don't depend on insulin to absorb glucose either. And they use a lot of it. More than the head. And in humans, typically the first place for glucose damage to show up is, yep, you guessed it, the feet. So I'm going to take a few minutes to talk about the possible role of glucose in the pathogenesis of laminitis and suggest a couple of ways glucose might cause laminitis or at least contribute to its development or severity. The story starts with advanced glycation end products, AGEs. AGEs are formed when glucose binds irreversibly to a protein. They accumulate with normal aging at an extremely accelerated rate under obesity-related insulin resistance. Obese humans with metabolic syndrome have markedly higher blood levels of AGEs than obese individuals who don't have metabolic syndrome. So the elevated AGE levels are associated with metabolic syndrome, insulin resistance, and not with obesity itself. I assume the same is true for horses. The thing about AGEs is that they interfere with the function of anything they bind to. And one of the most prominent things they target is collagen. Collagen is the most abundant protein in the animal kingdom. It provides structure, strength, and support to connective tissues throughout the body. And among other things, collagen plays a vital role in the health and structure of blood vessels. It provides structure and strength to blood vessel walls, and it helps maintain elasticity and flexibility. And because collagen is a stable protein with a very long half-life, these glycated proteins accumulate, irreversibly thickening the walls of blood vessels, interfering with the delivery of nutrients and other vital substances to tissues, and with the ability to adjust blood flow when appropriate through dilation and constriction. And guess what? Morgan and her colleagues found vascular dysfunction in laminitic horses. And because the dysfunction occurred in facial tissue as well as hooves, they concluded the vascular dysfunction in the hooves is likely to be due to the underlying metabolic disorder, insulin resistance, rather than secondarily to laminitis-induced damage to the hoof. This is a very important finding, it seems to me, in part because hooves are highly vascularized, as you can see. So does the vascular dysfunction reported by Morgan contribute to the risk of obesity-related laminitis? I don't know, but it seems possible. If so, it's reasonable to suggest that the culprit is likely to be AGEs formed from prolonged exposure to elevated glucose levels. Prolonged exposure, not just the 3 days of infusion used in the earlier study I mentioned before. One more thing about collagen. AGEs impair the mechanical properties of collagen. And there's a ton of collagen in hooves. For orientation, this is the hoof wall, and this is the coffin bone. This is the general area where attachments fail during laminitis. The sublamellar dermis is composed of collagen bundles that send filaments that attach to the basement membrane. The basement membrane is a sheet of extracellular matrix that forms the interface between the lamellar epidermis and the dermis. It's a three-dimensional sheet of fine, collagen filaments. According to Michelle Visser and Chris Pollitt, basement membrane changes are the first event in lamellar failure during laminitis. So as I said, there's a ton of collagen in the hoof, and it's integral to the structures that fail during laminitis. In fact, this whole area is composed principally of collagen. So collagen provides mechanical strength to tissue. The question is, does hoof collagen become glycated when a horse has metabolic syndrome? Remember, glucose doesn't require the help of insulin to get into hooves, and non-insulin mediated organs get flooded with glucose under insulin resistance. So do resulting AGEs compromise the strength of all those collagen structures between the hoof wall and coffin bone? I don't know. As I said, the possibility that glucose might cause the damage in horses as it does in humans hasn't been addressed beyond the superficial. So what's the point of this conjecture about glucose? Well, in my opinion, the potential importance of glucose to the development of obesity-related laminitis has been prematurely dismissed, particularly since glucose, not insulin, is directly responsible for the damage insulin resistance causes in humans. And it's a slow process. It takes time for the damage to occur. Think about this. What do most people do? What do veterinarians advise people to do when their horse is overweight and headed toward insulin resistance? Cut back on calories, right? Feed less and the animal will lose weight and the risk of insulin resistance and laminitis will be reduced. The problem is cutting back on calories comes with all sorts of serious problems, which I've described in the Gateway Clinic. On the other hand, what if the focus is on glucose? Why is glucose important to the body? It's converted to ATP, which provides the fuel that supports the processes of life. It's the gas that drives the cellular engine. So maybe rather than focusing on calorie restriction, we should focus more on burning the extra fuel. Use up the excess glucose hanging around in cells and the bloodstream. And in fact, exercise is by far the #1 antidote and preventative for insulin resistance, and it carries no harmful side effects like calorie restriction does. And you can easily find real-life examples. Sumo wrestlers are one. Sumo wrestlers are obese but not insulin resistant. Why? Because they exercise religiously. And with that, I'll stop. Thanks for listening.